Hi, this is Miss Conley, and we're going to go over the absolute basic structures of how a lab report is set up for MYP Science. It's very prescribed. You have to have certain things, and they have to be done in a certain way. Um, it's so that we can use the rubrics and the criteria, which is criterion B and criterion C. Don't ask me why it's not A and B. I don't know. But criterion B and C. And to follow through that, I've got this pretty strict and rigid type format that you have to follow, but it's for your own good, and it helps you look at your science investigation very systematically. We've talked a lot about being systematic when we're being a scientist, so this kind of goes along with that. So the basic lab report layout is this. I purposely made sure that you couldn't read the words because the words are different for every lab report you do. The structure is the same, okay? So this is basically, you've got your title, and I will explain in future videos what an RQ is. It's a research question. And I'll explain what a hypothesis is, and I'll explain the design portion, which is an independent, dependent, and control variables, which there are two. I'll explain all of this, but then you have to have an equipment list and procedures, and then you have to have a table and a graph. This isn't always a line graph, it could be a bar graph, and then a conclusion and evaluation, and then listing any sources that you cited, okay? You have to have all of those components, and they have to be done in a certain way. It's not because I'm mean, it's because we need to get you into a very scientific manner of looking at problems. So, when to do what? In a timeline of a whole lab report process, we're looking at 2 to 3 weeks, okay? It's a 2 or 3 page document when you get done. If you were to print it off on the copier, it would be, 2 to 3 pages, not double spaced. And the whole first part here, this bit from the title, research question, hypothesis, all the way down to and including your procedures, is all done before you even start doing your experiment. Because you have to do the planning. Planning is very important. If you don't do the planning, you end up running late and having to redo things, and then your data's all messed up, and it's just a big, huge problem. Plus, planning is important. So you have to do all of this stuff before you've even started doing the experiment. During that process, since we will put this on a Google Docs in a shared mode, I can go back and I can look at all of these things that you've got and give you feedback on them. And until I say lab reports are due, we're done, you can go back and take all that feedback and make any improvements that I ask for. Those people who don't go back and make improvements, I get annoyed with, because why am I doing the feedback if you're not going to use it, okay? So for this, you have to do all of this stuff before we do the experiment. Then we have to have some kind of little time to work out the bugs from your experiment, because up until this point you've planned it and you say, okay, I think I'm going to do this, and then I'm going to do this, and then I'm going to do this, but there might be flaws in that. You might have to change the amount of water you're going to use, or you might have to change the time you're going to allow something to happen. So this working out the bugs thing is a couple of days here and there, and this is when you figure those things out. And then you go back and you edit. Please do not forget to go back and edit. Please, you have to go back and edit. Remember, I'm not going to grade it until it's all done. You have every right to go back and edit everything until that date. So you work out all the bugs. Then all of this stuff is done afterwards, because you do the experiment. That's the then collect data part. So you work out the bugs, then you collect the data. And then you go back to your document and you put in your table, and we'll see how those go in future videos. And you do your graph, and you do that. You'll see that in future videos, and it's not always a line graph. It might be a bar graph. Then you do a conclusion, which everybody messes up the conclusion because there's so much to it. So you have to do the conclusion with everything that's being asked for, the evaluation section, and then any sources that you've referenced. I can try to go back and give some feedback on this section, but generally the time frame is a little too tight after people are done with their data collection. So during this time frame, it's probably best for you to come to me directly in class and say, hey, is this right on my table, or is this the right graph I'm supposed to be using? If I told you a million times, I might huff, but I'll still tell you, or at least point you in the right direction, okay? So all of this stuff has to be done after you've done your experiment. All right, time-wise, this here, the planning for it is about a week because you have to research your idea. You have to get some idea of what science out there says is going to happen. And then you got to do all this writing. So this stuff is all done about a week's worth of classes. Let's say we meet 3 times in a week. Then it'll be 3 class periods that we use to do this. But we'll have about a 1-week calendar time to do this. The working out the bugs and then the data collection. Working out the bugs is like a day, but then the data collection, I usually reserve 2 days in class for you guys to do the actual data collection for your experiment. That, some people won't be working in class though. Some people have an experiment that's just by nature something that they can't do in the lab and they have to do it like at home. So bear that in mind. It's okay. I don't mind if you do some work at home instead of here in the lab, as long as you do it. So we're looking at, for working out the bugs is like a day, but then we've got a day or 2 for your data collection. So we'll plan that out. And then after you've collected your data, to put everything in the right form and finish everything up, make any changes or corrections or improvements that you want to make, that's going to be another week too. All right, so what this all has to do with the MYP criteria. This whole section here is what we call experimental design. That's all your planning, that's how you set it up and everything. That's all part of the MYP criterion B. And then you do your experiment and everything. And then you have the second part. This all here is how you analyze your results, and that has to do with criterion C. So this is where we get the score for criterion B. And this is where we get the score for criterion C. That's how it all works out for the structure of a lab report. And then what you need to do from there is we need to go back and go to the top, look at each little part, like the research question and the hypothesis and blah, 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 blah. And we'll pick all of those apart. This was just to give you a good overview of how the whole thing works, okay? So I hope that helps. If you have any questions, of course, you can ask, but I think I pretty much covered everything, I hope. And the next video you will want to do is independent, dependent, IV, DV, RQ, and HYP. Basically, it's, look, you can search for Jennifer Connelly, IV, DV, RQ, HYP. And that will take you to the independent variable, dependent variable, because you have to figure those 2 out first, and the research question or hypothesis. So that'll start in detail, okay? So as soon as you're ready, you can go to that video, and this is the basics of what your lab reports look like. You will know this like the inside of your eyelids by the time we get done with 8th grade. Okay, have a good day. Bye-bye.